Welcome back to the deep dive. Today we are diving into genetic engineering.
Yes, and I mean, this is such a massive topic.
It really is.
Hmm.
Honestly, looking at our sources today, it feels like the most profound thing we've ever talked about.
Oh, absolutely.
We've got everything from old Supreme Court transcripts from the 80s all the way to like brand new 2026 clinical trial data. Oh, and environmental reports from the jungle.
It's a huge stack of information, but our main mission here is really to track how we went from just, watching traits passed down in a garden to literally rewriting the biological code of life.
Right. Curing diseases, changing the environment, maybe even redesigning ourselves.
Yeah, it's the ultimate pivot for humanity. We're going from just being passive observers of nature to actually being the authors of it.
So, to kick this off, I want you to picture walking into this dusty, forgotten attic.
Okay, I'm picturing it.
Sitting right in the middle of the room is this massive, heavy, leather-bound book. You open it up and you realize, wait, this isn't just a book. It's the literal instruction manual for building you.
Every single detail.
Exactly. Your eye color, your height, your laugh, everything.
It's basically the most complex library in the universe just sitting there on the page.
Right. And for all of human history, we couldn't even get the cover open. But then we finally learned how to read it.
Which was a miracle in itself.
But here's where it gets crazy. Imagine if you suddenly noticed a pen, an eraser, and a pair of scissors sitting right next to the book.
The moment you realize, oh, this ink isn't permanent, we can actually edit this.
Yes. But to understand how we are editing the manual right now in 2026, we have to kind of back up. Where does this whole story actually start?
Well, to really lay the foundation, we need to go back to the 1860s. So picture this Austrian monk, Gregor Mendel, just hanging out in his monastery garden.
Oh, right, the pea plant guy.
Exactly. He is obsessed with peas. He's cross-pollinating thousands of them, right? Smooth ones, wrinkled ones, white flowers, purple flowers.
Okay, wait, let me stop you right there. Why peas? Like, what did people even think was happening before he did all this painting pollen on peas?
So back then, people basically believed in what was called blending inheritance.
Like mixing paint?
Yeah, exactly. They thought if you had a tall parent and a short parent, the kid would just naturally be medium height, like a smoothie of traits.
Okay, so a total blend.
Right. But Mendel picked peas because their traits don't do that at all.
Yeah.
A pea plant is either tall or short, it's green or it's yellow. There's no yellowish green in between.
So they were the perfect way to prove the whole smoothie theory wrong.
Precisely. And he discovered this amazing mathematical rule. When he crossed a pure yellow pea plant with a pure green one, the babies weren't blended. They were all yellow.
Wait, so the green just vanished?
It looked like it did, but then he took that all-yellow generation and crossed them with each other, and suddenly, boom, the green peas came back in the next generation.
Oh, wow.
Yeah, and they came back in a very specific, perfectly predictable ratio.
Yeah.
Exactly 3 yellow peas for every 1 green pea.
So the green instructions didn't actually disappear. They were just like hiding in the background.
Exactly. Mendel figured out that traits are passed down in these invisible separate units, and some are loud, like a loud voice in a room, and some are like a whisper.
Right. Dominant and recessive.
Yeah. If the loud voice is talking, you don't hear the whisper, but the whisper is still there. The traits do not melt together at all.
So he basically proved that the instruction manual is made of individual sentences that don't change, even if you couldn't see the actual words.
Exactly. It took another 100 years for scientists to actually see the physical shape of the manual, which brings us to the 1950s.
That's the famous twisted ladder, right?
Yes. Scientists bounced X-rays off the molecule and saw that the code of life is shaped like a twisting spiral staircase, the double helix.
And the rungs of that ladder are made of just 4 chemical letters, right? A, T, C, and G.
Yeah, just those 4.
Which is wild to me. Like a mushroom, a whale, a human, a bacteria, we're all written using the exact same 4 letters.
Every single living thing on Earth. Just a 4-letter alphabet. And the really genius part is how the letters pair up. A always pairs with T, and C always pairs with G.
Always.
Always. So if you pull the ladder apart down the middle, each half acts like a perfect template to rebuild the other half. That's how life copies itself.
Okay, that is super elegant. But, so we figure out the shape of the ladder, we're reading the book. How do we actually start cutting the pages?
That leap happens in the 1970s. Scientists were looking at this microscopic war going on between bacteria and viruses.
Right, because viruses hunt bacteria.
Yeah, they inject their own code into the bacteria to hijack it. But scientists noticed the bacteria had a defense mechanism. These special proteins that act like, well, Molecular scissors.
Built-in scissors to chop up the virus.
Exactly. The scissors scan the virus's twisted ladder looking for a very specific sequence of letters. And when they find it, snip. They cut the ladder right in half.
Wow. So when did scientists realize they could just, steal those scissors for our own projects?
Pretty much immediately, they realized that since all life uses the same alphabet, those scissors would cut any DNA, not just virus DNA, human, plant, anything.
But if you cut it, you also need glue, right?
Yep, and nature had that too. When the scissors cut, they usually leave a little sticky overhang. Add in a naturally occurring biological glue, and you can paste two totally different pieces of DNA together permanently.
Okay, let me make sure I'm getting this analogy right. So if the code of life is this giant encyclopedia, we went from just reading it in the library to taking literal scissors, cutting a paragraph out of a bird book, and gluing it into a bacteria book.
That is exactly what we did. And because it's the universal alphabet, the bacteria just reads the bird paragraph and follows the instructions. It doesn't know the difference.
That is insane. What was the first thing they actually used that for?
A huge one was human medicine in the late 1970s, specifically for diabetes. Before this, people with diabetes had to use insulin that was harvested from pig and cow pancreases at slaughterhouses.
Oh, that sounds incredibly inefficient.
It was terrible. It took like 8,000 pounds of animal glands just to get 1 pound of insulin. Plus, animal insulin sometimes caused allergic reactions.
So they used the scissors and glue.
Yeah. Scientists went into a human cell, cut out the paragraph that makes human insulin, and glued it straight into a simple bacteria's instruction manual.
But wait, wouldn't the bacteria look at human code and just reject it? Like it's a totally foreign language.
No, because of that universal alphabet. A C is a C, whether you're a human or a bug. The bacteria just read the new instructions and started pumping out perfect 100% human insulin.
So they basically turned the bacteria into a tiny medicine factory.
Exactly. And bacteria multiply super fast. Suddenly, we had endless supplies of pure human insulin. It changed millions of lives.
Which is amazing, but it also brings up this huge dilemma. If you just invented a brand new living thing, like a bacteria that makes human medicine, who owns it?
Right.
Like, can you patent a living creature the way you patent a toaster?
This was a massive turning point. Our sources talk about a landmark Supreme Court case in 1980. A scientist had created a custom bacteria designed to eat crude oil to help clean up ocean spills.
Oh, sounds like an environmental miracle. Did he try to patent it?
He did. And the U.S. Patent Office said absolutely not. They told him, you can't patent a product of nature.
Because you can't just find a new bird in the woods and patent it. You didn't invent the bird.
Right, but the scientist argued, I didn't find this bacteria, I invented it. I used tools and editing to make something that never existed in nature.
And what did the Supreme Court say?
In a super tight 5-4 decision, they sided with the scientist. They said, you can patent anything under the sun that is made by man, even if it's alive.
Well, 5-4. One vote basically changed the entire global economy. Because suddenly there's a huge financial reason to edit life.
Exactly. Billions of dollars flooded into the industry overnight. Biology became a massive business.
And it didn't take long for this to hit the grocery store, did it?
Yep.
We read a lot about agriculture in the 90s.
Yeah, the first engineered whole food was a tomato in 1994. They tweaked its instructions so it wouldn't get mushy. It stayed fresh on the shelf for weeks.
A super tomato. Was it a hit?
Commercially, no, it flopped. It tasted pretty bad and was too expensive. But it opened the door for staple crops like corn and soybeans.
Right. This is the whole genetically modified food boom. What did they do to the corn?
They gave it a gene from a naturally occurring soil bacteria. This gene makes a protein that is totally toxic to specific insect pests, like caterpillars that eat the corn.
Wait. If the corn is producing its own toxic bug spray, how is that safe for us to eat?
It's actually brilliant. The toxic protein only activates in a highly alkaline stomach, which caterpillars have. Humans have super acidic stomachs. So to us, the protein just breaks down like normal food. It's completely harmless.
Oh, wow. So the plant defends itself, and the farmer doesn't have to spray chemicals everywhere?
Exactly. They also made crops that were totally immune to powerful weed killers, so a farmer could spray the whole field, kill all the weeds, and the crop would just be perfectly fine.
Okay, so you give the crops these literal superpowers, which sounds great, but there's a massive public backlash, right? People were terrified.
Yeah, and a lot of the fears were totally valid. Ecologists were worried about super weeds, Like, what if the immune crop cross-pollinates with a wild weed and suddenly we have unkillable invasive weeds taking over forests?
That is a terrifying thought.
There is also huge concern about corporate control. Because these seeds were patented, farmers weren't legally allowed to save their seeds for the next year. They had to buy brand new, expensive seeds from the corporation every single season.
So it completely changed how farming worked.
It really did.
Okay, so we have this era of cut-and-paste biology. But from what I'm reading in our 2026 sources, those early molecular scissors were actually kind of clumsy, right? Like using a meat cleaver for brain surgery.
That is the perfect way to describe it. The old scissors only looked for short, 6-letter sequences. In a human instruction manual with 3 billion letters, a random 6-letter sequence repeats thousands of times.
So you try to fix one typo, but the scissors accidentally chop up the book in 40 other places.
Yes, and worse, when you completely snap the DNA ladder in half, the cell panics. It knows a broken ladder is lethal, so it just furiously jams the pieces back together, making a ton of mistakes and causing random scars in the code.
Okay, so if we actually want to cure complex diseases, we can't rely on a panicking cell. We need something sharper.
Which brings us to the precision era in the 2010s. Scientists found a much better tool in bacteria, the one we've all heard of in the news.
The famous search and cut tool.
Right. It's basically programmable scissors with a highly specific GPS guide attached.
How does the GPS part work?
Imagine giving your scissors a custom microscopic piece of tape with an exact 20-letter sequence on it. The tool travels along the 3 billion letters, unzipping the DNA and testing that tape. It only makes a cut when it finds a perfect 100% match.
And since a 20-letter sequence is so long, it's pretty much guaranteed to only show up once in the whole book, so you don't get all those accidental cuts.
Exactly. It was a massive Nobel Prize-winning leap. But it still had one major biological flaw. It still snapped the ladder completely in half, which could still trigger that cellular panic.
So even with the GPS, it's still a pair of scissors. Which brings us to the cutting edge of 2026. How do we edit the manual without tearing the page out?
That is where we get the new word processor tool. It's called prime editing.
Okay, break this down. Why is it a word processor?
It uses the same brilliant GPS guide to find the exact typo. But the scissors are intentionally dulled. Instead of cutting the whole ladder in half, it just gently nicks one side of it. The backbone of the ladder stays structurally totally intact.
So the cell's alarm system doesn't go off, it doesn't panic.
Right. And then a second machine built into the tool activates. It acts like a microscopic pen. It literally reads a little template it brought along, erases the bad letters, and pencils in the newly corrected letters right into the nicked strand.
Wow, so instead of tearing a page out and furiously taping a new one in, we're just gently erasing one misspelled word and penciling in the correct spelling.
That's the exact mechanism. And in 2026, the capabilities are staggering. It can fix all 12 possible types of single-letter typos in the human code.
And a lot of awful diseases, like sickle cell anemia, are caused by literally just 1 or 2 wrong letters out of 3 billion, right?
Absolutely. One typo changes a protein and causes a devastating disease. And this tool can even insert whole missing paragraphs or delete extra ones. It's pure precision.
It honestly sounds like magic. But I have to push back here. If we have this perfect biological word processor, and we can just pencil in the cure without causing a panic, why aren't we curing every genetic disease tomorrow?
That is the million-dollar question. And it's the biggest roadblock in all our 2026 data. It's called the delivery dilemma. The tool is perfect, but physically, it's just enormous at the microscopic level.
Wait, what do you mean it's too big?
Think about the logistics. If you want to fix a disease in someone's liver, you can't just hand them a pill. You have to transport millions of these microscopic word processors into their bloodstream, navigate to the liver, and somehow force them inside the heavily defended liver cells.
Right, because our cells are designed to keep foreign things out. You need a delivery truck.
Exactly.
Yeah.
Scientists mostly use 2 types of delivery trucks. The first are harmless, hollowed-out viruses. Because viruses are absolute experts at breaking into cells, scientists just scoop out the bad viral DNA and stuff the editing tools inside the empty shell.
Like a biological Trojan horse.
Exactly. The other truck is basically a microscopic fat bubble. Since our cell walls are also made of fat, when the bubble bumps into a cell, they just merge together like 2 soap bubbles, dumping the tools inside.
Okay, those both sound like great systems. What's the issue?
Cargo space. The older, clumsier scissors fit perfectly into the hollowed-out viruses, but the new word processor has the GPS, the nicking scissors, and the pen all fused together. It's just too bulky. It literally doesn't fit in the delivery truck.
Oh, man. This is like ordering this massive, amazing sectional sofa online, but when the delivery guys get there, your front door is way too small. The sofa's great, but it's useless sitting on the lawn. You either need a bigger door, or you have to ship the sofa in smaller boxes and build it yourself in the living room.
That is the exact analogy, and that's exactly the workaround scientists are using. It's the system called split-entanes.
Split-entanes. How does that work?
They actually split the massive word processor blueprint directly in half. They package the first half into one hollowed-out virus and the second half into a second virus.
Right.
When both viruses break into the patient's cell, they release their halves, and these halves have special chemical magnetic clasps on them. When they bump into each other inside the cell, they instantly snap together, building the full word processor.
That is completely unbelievable. We are doing microscopic flat-pack furniture assembly inside a living human cell.
It is a massive biological feat, and when they finally get the delivery right, the results are basically miracles. The 2026 clinical data is showing things that were pure science fiction 10 years ago.
Tell me about some of these miracles. Like, what does this actually look like for a real human being in a hospital bed?
Take severe blood diseases like sickle cell anemia. Patients have a single-letter typo that makes their red blood cells rigid and sickle-shaped. They jam up in the blood vessels, causing agonizing pain and organ damage.
And before this, they just had to manage the pain and get constant blood transfusions, right?
Yeah, that was it. But now we have functional cures. Scientists take the patient's blood stem cells out of their bone marrow, they fix the typo in a lab using the tools, and then put the cells back into the patient. The patient just starts making perfectly healthy, round, red blood cells.
They're just cured.
They're living totally pain-free lives.
That's incredible. And I also read something about a treatment for a skin disease.
Yes, a devastating genetic condition that causes severe skin blistering. The patients are missing the instructions to make a specific biological glue that holds their skin layers together. Even light friction causes massive blisters.
How on earth do you edit the skin over someone's entire body?
They made a rub-on gel. The gel has the corrected instructions packaged inside deactivated viruses. Doctors just apply the gel directly to the patient's wounds, and the viruses deliver the instructions straight into the damaged skin cells.
And the cells just start making the missing glue.
Exactly. The wounds finally heal. Now, since skin sheds, it isn't permanent, but they can just reapply the gel whenever needed to keep the skin intact.
Wow. Okay, listening to this, it feels like we are in a golden age. We're curing blood diseases. We have gene editing skin gels. But your tone earlier when you said the clinic versus reality, it felt a little grim.
Right.
What's the actual reality in 2026?
The grim reality is this massive financial and logistical wall. Despite these absolute miracles, very, very few people are actually getting them.
Really?
Look at the numbers. There's one major company with multiple life-changing genetic therapies approved. But globally, only a few dozen patients have actually successfully gone through the whole treatment process.
Wait, a few dozen? If the cure is real and it's approved, why isn't every patient getting it?
Cost and complexity. These customized therapies cost somewhere between $2 to $3 million per patient. And it is not just a pill you pick up. For the blood diseases, the hospital process is brutal.
Right, because they have to take your bone marrow out and put it back.
Yes. And while the cells are being edited in a lab, the patient has to get intense, toxic chemotherapy to wipe out their remaining diseased bone marrow just to make room for the new cells.
Oh, wow.
During that time, they have zero immune system. They need weeks in highly specialized sterile isolation units with round-the-clock intensive care. Very few hospitals can even handle it safely. The healthcare system is just completely paralyzed by the millions of dollars it takes for one single person.
That is such a tragic irony. We've finally built the ultimate tools to cure the incurable, but our bank accounts and our hospital infrastructure are completely failing the patients.
Exactly.
And from the sources, it's not just the back-end costs at the hospital, right?
Right.
Something is happening with the front-end research funding, too.
Yes, the geopolitical and economic climate in 2025 and 2026 has been a massive shock to the system. And just looking at it neutrally, it is heavily dictating how fast the science can move.
What's actually happening in the data?
We're seeing historically unprecedented funding cuts by the U.S. government. Billions of dollars have been suddenly pulled from biological research. Thousands of grants have just been suspended or terminated.
So what does that actually mean for the scientists? Just slower paperwork.
No, it's immediate and catastrophic. It halts clinical trials in their tracks. The labs trying to invent the next generation of tools, maybe the tools that would make these therapies cheaper and easier to deliver, they suddenly have no budget to buy chemicals or pay their staff.
So the labs just shut down.
Many do, and it's triggered a severe brain drain. Top researchers are seeing their money evaporate, so they're packing up and moving to countries in Europe or Asia that are aggressively funding this research.
Man, so we finally mapped the manual, we build the perfect word processor, and right at the finish line, we run out of money, we run out of hospital beds, and we lose the scientists. That is a lot to sit with.
It really is the critical bottleneck of our time.
I want to pivot the conversation a bit here, because this creates a very interesting and honestly kind of dark tension. We have all these hurdles in the clinic trying to cure sick people, but the tools themselves are getting so much more accessible. What happens when people look past curing diseases and start thinking about enhancement, like designing the perfect human?
The ethics of human enhancement. Yes, we absolutely have to talk about this. To understand 2026, we have to revisit this highly controversial event from 2018. It was the moment a massive line got crossed.
You're talking about the CRISPR babies, right?
Yes. A scientist named He Jiankui illegally altered the genetic code of twin girls when they were just single-celled embryos in a petri dish. He used the search and cut tool to try and make them genetically resistant to HIV.
Okay, wait. We just spent 20 minutes celebrating how amazing it is to edit blood cells and cure diseases. Why was the whole scientific world so universally outraged when he tried to make kids immune to a virus?
Because of what stage of life he edited. When you edit a living patient's skin or bone marrow, that edit stays inside that patient. If they have a child, the child inherits the original unedited DNA.
Right.
But when you edit an embryo, the foundational cell of a new human, you are doing germline editing. As that one cell divides into 2, then 4, then a billion, that edit is permanently copied into every single cell of those girls' bodies.
So it's everywhere.
Yes, including the reproductive cells. Which means those edits and any accidental scars the clumsy scissors might have made will be passed down to their children and their children's children forever. He unilaterally altered the genetic lineage of the entire human species.
Wow. So he essentially took a permanent marker to the instruction manual of future generations without their consent, and without even knowing the long-term consequences.
Exactly. He went to prison, and editing embryos for pregnancy is now strictly banned globally.
But the desire to design a child didn't just go away, did it?
Not at all. While editing embryos is banned, complex screening of embryos is an absolutely booming industry in 2026. Sociologists are actually calling it consumer eugenics.
Consumer eugenics? That sounds incredibly dystopian. What are people actually paying for?
Well, fertility clinics used to just screen embryos to make sure they didn't have a fatal single-letter disease, which is widely accepted as a great way to prevent suffering. But now they're doing these complex full genome scans.
To look for what?
They're trying to predict highly complex traits, lifelong risks for heart disease, diabetes, or even controversial things like height, eye color, or intelligence.
Hold on, let me push back on this. Are we really treating human life like customizing a video game character? Like, give me the tall one with a +10 to the intelligence stat. Scientifically, intelligence is based on a million different things, right? Aren't these stats basically just guesses?
That is exactly the massive scientific concern. Things like intelligence or heart disease aren't controlled by one simple sentence in the manual. They're influenced by thousands of different genes, all interacting in ways we barely understand.
So you can't just isolate the smart gene.
Right. The clinics use these huge databases to find tiny statistical correlations, and they generate a probability score for the embryo. It's purely a statistical guess. It completely ignores the environment the child grows up in, their diet, their education, and just random chance.
So wealthy parents are paying thousands of dollars to pick embryos based on an algorithm's guess, which could be totally wrong.
Yes. And the backlash is already here. Families are actively suing these testing companies right now, claiming the clinics gave them overly confident predictions, which led them to throw away perfectly healthy embryos just because a computer guessed they might have a higher risk of high cholesterol in 40 years.
That's profoundly disturbing. It's turning fundamental human traits into luxury products. If only wealthy people can afford to screen their embryos for the highest scores, what happens to the rest of society?
That exact fear has started a massive political movement. Legal scholars and civil rights advocates are pushing for a federal law called the Genetic Equity Act.
What would that do?
It tries to establish a brand new human right, the right to a random genetic code. They want ironclad laws so employers or health insurance companies can never demand to see your embryo probability score and use it against you.
To prevent a biological caste system.
Exactly. They want your life to be dictated by your actions, not by the genetic prediction score your parents bought before you were born.
Okay, let's look at the board. We have the tools to cure diseases, but we can't pay for the hospital stays. We have the tools to screen human traits, but we're risking a biological caste system. We're clearly struggling with the ethics of editing ourselves. What happens when we take these powerful tools out into the wild? Because we aren't just looking at humans, right?
Not at all. The environmental applications happening in the wilderness are just as powerful and honestly even more controversial.
Let's get into this. How are we rewiring the wilderness?
The most potent tool right now is called a gene drive. And to understand how radical it is, we have to go all the way back to Mendel and his piece.
The 3-to-1 ratio.
Right. Mendel taught us that normally a gene has a 50-50 chance of being passed down. A biological coin flip, half from mom, half from dad.
Makes sense.
But a gene drive is a biological fast pass that completely destroys that rule. Scientists physically build the microscopic search and cut machinery directly into the animal's DNA.
Wait, they put the scissors inside the animal's reproductive code?
Yes. So when a modified animal mates with a normal one, the embryo gets one modified half and one normal half. But immediately, the scissors wake up, reach over to the normal half, cut it, and force the cell to copy the engineered trait over.
Whoa, it edits the other half of the DNA in real time.
Exactly. So the engineered trait is now on both sides. When that animal grows up and has babies, it has a 100% chance of passing it down, not 50-50, 100%.
So it completely overrides natural selection. If 100% of the babies have it, and they pass it to 100% of their babies, that trait would spread like wildfire through an entire wild population.
It's an exponential biological chain reaction.
I can totally see how powerful that is.
Yeah.
But why would we want to force a trait through a whole wild population? Give me a real-world example.
The biggest one happening right now is a multi-million-dollar project in Equatorial Guinea with mosquitoes trying to stop malaria.
Okay.
They engineered mosquitoes with a gene drive that targets female reproduction. They release modified males into the jungle, they mate with wild females, and the fast pass ensures all the offspring get the drive. Within a few generations, all the females become totally infertile.
Wow.
The mosquito population crashes, and the malaria transmission just stops.
That is staggering. You release a few mosquitoes, and in a few seasons, the local population is wiped out, potentially saving millions of lives.
Right.
Are they using this for anything else?
Ecological conservation, actually. Like invasive mice and rats that were accidentally brought to remote islands by ships. They eat the eggs of endangered seabirds and destroy the ecosystem.
So instead of dropping toxic rat poison all over an island.
Right. They are working on releasing gene drive mice. The drive quietly spreads infertility through the invasive mice. They stop reproducing, and the island recovers. No poison needed.
But wait, that brings up a terrifying risk. Say you put these modified mice on an island off New Zealand. What if one modified mouse sneaks onto a cargo ship, escapes to the mainland where that mouse is actually native and important, and the drive spreads? Couldn't one escaped mouse trigger the extinction of a whole species globally?
Welcome to the extinction debate. That is the exact nightmare scenario that keeps ecologists awake. A gene drive doesn't know it's supposed to stay on an island. Once you release a biological fast pass, it is almost impossible to recall it.
Have global leaders talked about this? What's the consensus on risking a planetary-scale disaster?
In late 2025, there was a massive, highly contentious global vote at the International Union for Conservation of Nature. They debated a total, absolute ban on releasing any gene drive into the wild.
And did they ban it?
They rejected the ban. They basically decided that the global biodiversity crisis is so severe we can't afford to take any potential tools off the table.
So the risk of doing nothing is worse than the risk of the gene drop.
Exactly. They opted for extreme caution instead of a ban. Rigorous lab testing, intense community engagement, phased evaluations. They chose the difficult path of heavy regulation over total paralysis.
Man, it's a massive calculated gamble with the planet. We are literally the engineers of the biosphere now. And just to add one final layer of craziness to this deep dive, human scientists aren't even the only ones designing these tools anymore, right? What is happening with AI and biology?
This is the newest frontier,
Huh.
and it's accelerating everything we've talked about by an order of magnitude. Artificial intelligence isn't just analyzing data anymore, it is acting as a biological architect.
What does it actually mean?
For decades, the biggest mystery in biology was how proteins fold. Proteins are the machines of life, the scissors, the delivery trucks. They're made of chains of amino acids, but the 3D shape they fold into determines what they do.
Like Rosalind Franklin figuring out the twisted ladder shape.
Yeah, and figuring out one 3D shape used to take years of slow, painful lab work. If you wanted a new tool, you had to go dig in the dirt and hope you stumbled across something useful that evolved naturally.
So how did AI change that?
A few years ago, AI models ingested massive databases and learned the actual physical rules of how amino acids interact. Now, an AI can instantly predict how a sequence will fold into a 3D machine.
Saving decades of work.
Yes. But in 2026, it went a step further. Scientists can now just type a prompt into an AI. They say, design me a brand new delivery truck protein that bypasses the immune system and only opens up inside a liver cell.
And the AI just invents it. A protein that has never existed.
It simulates millions of years of evolution in a few hours. It designs a brand new tool from scratch, tells the scientists the exact DNA code needed, they synthesize it, and a cell builds a machine that never existed on Earth. It's cutting development time in half, creating tools far more precise than anything we could ever find in nature.
So if I had this right, AI is acting like a super fast architect, designing brand new biological Lego blocks that never existed. We're using those blocks to build flat-pack word processors, loading them into custom fat bubbles, and fixing single typos in a human cell. And we're using similar tools to build fast passes to redesign the whole planet's ecosystem.
That is flawlessly accurate. We have fully integrated digital computer code with biological DNA code. The feedback loop between AI and genetic engineering is driving a revolution faster than anything in human history.
This has been absolutely staggering to process. You know, for billions of years, life on Earth was strictly by random chance, by tiny mutations and the unfeeling grind of natural selection. If a typo happened, nature just decided if you lived or died. But now, the pen is firmly in our hands. We are no longer just the products of evolution. We are the architects of our own evolution.
We have completed the transition. We really are the authors now.
And the ultimate question we face now isn't whether we have the power to rewrite the book of life. We clearly do. The question is whether we have the wisdom to know which pages we need to edit and which pages we must absolutely leave alone. Thanks for coming along on this deep dive with us. Remember to follow us on your favorite podcast platform, and see you on the next dive.